Hi, my name is Naoyuki Kamatani, the CEO of Taka Pharma Company. We are now developing a very promising new drug, which is in a clinical stage. So please listen to me. I talk about ATP or energy enhancement for the treatment of Parkinson's disease, dementia, and mitochondrial disease. Just as energy is a critical issue for the planet, it is also a critical issue for the human body. Mounting evidence suggests that Parkinson's disease, Alzheimer's disease, and mitochondrial disease are caused by cellular energy shortage. But no disease-modifying drug is available for Parkinson's disease, no effective oral drug is available for Alzheimer's disease or vascular dementia, and no effective drug is available for mitochondrial disease either. An AI study from 5 US universities to analyze a vast amount of data, including GWAS, omics, and others, identified febuxostat as the most effective drug for Alzheimer's disease, and huge clinical data from 7 million patients confirmed the effect. In addition, dementia suppressing effect of XOR inhibitors has been reported from various other studies. In the left side panel, US researchers reported that allopurinol and febuxostat suppressed dementia in more than 30,000 patients. In the right side panel, Korean researchers reported that febuxostat suppressed Alzheimer's disease and vascular dementia to about 1/5 of the normal risk. A meta-analysis also confirmed the results. This is a list of papers reported from various countries which reported the dementia suppressing effect of XOR inhibitors. So the dementia suppressing effect of XOR inhibitors, including febuxostat, is for sure. However, before all of those reports, we patented them in a very clever way. Using our unique technologies, we discovered the effect and patented combination drugs, each containing an XOR inhibitor including febuxostat and inosine. This is because we clarified the mechanisms of Parkinson's disease from genetics point of view. If you look at the genes responsible for Mendelian Parkinson's disease and sporadic Parkinson's disease, about 30% of the genes are mitochondrial related, and 15% are ubiquitin proteasome related, and 10% are autophagy lysosome related. And the genes responsible for aggregated protein alpha-synuclein are only 5%. Therefore, very likely, ATP shortage due to mitochondrial dysfunction is the most upstream abnormality, which leads to the dysfunctions of ubiquitin proteasome and autophagy lysosome systems, and finally leads to the accumulation of aggregated protein alpha-synuclein. So the effects of therapies to reduce aggregated protein will be limited. Then how can we enhance ATP? ATP is composed of the AMP part and the high energy part. The AMP part is enhanced if hypoxanthine increases. Hypoxanthine can be increased by either XOR inhibition or inosine supplementation. If the AMP part is enhanced, the high energy part is also enhanced, since more glucose goes to the high energy part. This is a list of our 3 major patented combination drugs. Each of our drugs is a combination of an XOR inhibitor and inosine, and hypoxanthine or IMP can be substituted for inosine. The patents were approved in Japan, US, China, EU, and other countries. This slide shows the mechanism of action of our drugs. Since the target enzyme XOR is not expressed in the brain, XOR in the liver is inhibited, and hypoxanthine increases in the liver and blood. Since hypoxanthine crosses BBB, it enters the brain and enhances ATP. The combination drug febuxostat and inosine is better than febuxostat alone, since it further enhances hypoxanthine. Then we administered febuxostat and inosine to healthy subjects for 2 weeks and measured hypoxanthine and ATP in the blood, and ATP and hypoxanthine were indeed increased. This slide shows a synergistic effect and the reduced side effects by the combination of the two compounds. The left side panel shows an increase of hypoxanthine. When feverzostat and inosine are administered separately, there was a little bit increase in hypoxanthine. However, when they were administered together, there was a marked increase in hypoxanthine. In the right side panel, we showed a change of uric acid. Inosine caused a marked hyperuricemia and feverzostat caused a marked hyperuricemia. But when both of the compounds were administered together, the adverse events of both of the compounds cancelled with each other. So there was a synergistic effect and reduced side effects. Then we administered feverzostat and inosine to 26 patients with Parkinson's disease for 2 months. And the primary endpoint MDS-UPDRS Part 3 was significantly improved. In addition, we found a very interesting phenomenon. Hypoxanthine is very low in Parkinson's disease even before the treatment, but we found that our drug was more effective for Parkinson's disease with lower hypoxanthine. So we can use a companion diagnostics technology. The number of the patients with Parkinson's disease is about 1.5 million in US, Japan, and EU only. First, we target Parkinson's disease with low hypoxanthine, since a previous study confirmed that our drug is more effective for Parkinson's disease with low hypoxanthine. This is narrow the target of a drug, but will increase the success rate very much. When additional funds are obtained, we start the development of drugs for other diseases also. This is the management team. My name is Naoyuki Kamatani, the CEO of Takao Pharma. And I have authored over 600 papers, including 34 in Nature and Nature Genetics. Other members are also very reliable persons. This is a business plan and necessary expenses. We are going to start a phase 1B study from the end of this year, and we are going to enroll 24 Parkinson's disease patients. And we analyze the result very carefully and proceed either to a phase 2-3 combined study or to a phase 2 study followed by a phase 3 study. And the cost required until the end of the phase 1B study will be $5 million. And now we request $5 million to complete the phase 1B study. The end of phase 3 study will be in 2027, and the submission for approval will be in 2028, and the total cost required will be $50 million. This is the business model and the revenue model. The maximum sales of a drug will be in 2037, and the maximum sales will be about $270 million. As exit strategy, IPO or M&A is contemplated when POC for Parkinson's disease is obtained in 2025 or upon approval in 2029. The enterprise value in 2029 is estimated at $300 million for Parkinson's disease with low hypoxanthine and Japan, US only. Some of you may be concerned about the pricing of a drug because a drug is a combination of two very old compounds. But FDA recently approved Auvelity as a fast-acting antidepressant last August. Auvelity is also a combination of two very old compounds, but the retail price is pretty high, about $8,000 per year per person. We received the FBRI Award at the 3rd Kyoto University Life Science Showcase in San Diego in 2022. Please let us know if you are interested in our ATP enhancers. Thank you very much for your attention.